Good afternoon, ladies and gentlemen. Let me quickly start the presentation. Here we go. First, as a first slide, please, let me give you a short framing. This is the compound we are talking about. We call it Trivehexin, and Margaret has shown the structure before in her introductory slide. And this is sort of image you get out from the PET imaging. And on this slide, I also mentioned the target, which is called alpha V beta 3 integrin. And I'm totally aware that if I mention the term integrin, some people have some associations with that, which is associated with angiogenesis and tumor growth. But this actually applies to alpha V beta 3 integrin. This is a sort of image which you get with those agents which are around now for, let's say, roughly 2 decades. But this is not our topic today. We're talking about a different story. The story I'm talking about is alpha V beta 6 integrin, and it's not endothelial, it's expressed on epithelial cells, and it binds to the latent-associated peptide, which is covering TGF-beta, and it releases TGF-beta, and so the expression of alpha V beta 6 integrin is related to the effects of TGF-beta. So what does TGF-beta in a tumor do? And I will keep this very short. Elevated TGF-beta concentrations are generally correlated with tumor invasion and tumor metastasis. So we have a marker here for tumor invasion and metastasis. And again here, comparison alpha V beta 3, which is not what I'm talking about today. We have a biomarker for carcinoma malignancy. Having that said, you might be asking which carcinomas the beta 6 integrin is expressed in. And you can see there are 2 indications which are quite well known already from immunohistochemical studies, pancreatic and head and neck cancer. You can see nicely here that it's a typical pattern that you find in immunohistochemistry, that the integrin is expressed on the contact front between the tumor cells and the tumor-associated fibroblasts, but it's expressed on the tumor cells, not on the fibroblasts. And we are conducting at the moment a large study which is heavily supported by our collaboration partner, Radiopharm Theranostics, to figure out in which other entities the integrin is expressed. So this is ongoing work. I can give you some sneak images here. You can see we have in some cases of lung adenocarcinoma, non-small cell lung cancer, we have a really high expression, and we have an intermediate expression on another fraction of the cases. What we're currently doing is checking out ovarian carcinoma. And as we speak, this study is going on, so there are many more data coming soon. So from the scientific point of view, I think we are on a good track here. But my good friend Jakub Simecek, he would probably say, when science has come to a conclusion, this is where the real work starts. And at this moment, I would like to ask him to come to the podium and tell you something about our ongoing efforts to bring this asset into the clinics.
Thank you very much, Johannes. Good afternoon. Good afternoon, ladies and gentlemen. So as Johannes already shown, there is a huge topic he was working on last 10 years. It's not to be forgotten that there is the huge effort behind the results we can present today. And as we are a young company, since TRIMT was founded in the last year, it's also our part to tell the story how we are developing together these compounds. And if you observe the development of the compounds, it always looks like a nice fairy tale where you do some chemistry and then you have some preclinics and some images in the mouse models and then the patients. But between the mouse model and the patients and the broad use, there is a huge gap, which was a topic that was really making us busy the whole last year, because there is a lot to do, and there are a lot parts and a lot collaborations on the way to the daily practice in the clinic. So the question is how to bring these preclinical data to the patients on the daily basis, and of course how to bring these to the goal to have not only a great diagnostic for various carcinomas, but also a reliable therapeutic option for the patients after they are selected as eligible for the therapy. So we are building our efforts on the really broad collaboration network across the globe. So we are working together with more than 30 partners from both industry and academia. And to name just a few, from the on the academic level, there is really the development of the new compounds, new motifs, all the chemistry and optimization. We are very happy to work with our former colleagues from Technical University of Munich, where the compound was also developed. So we're happy that we can continue there, especially the preliminary preclinical works, mainly with the group of Professor Susanne Kossatz, who is making a great effort and helping us a lot on our way in the development of the new compounds. And then also we have very nice collaboration with the University Clinic in Dresden, which is on the other way, just around the corner for us, as we are a company from Radeberg near Dresden in Germany. We work also very, very well together with Chematec from France, which you probably know, and we are happy to have such a partner because they're supplying us with all the very specific chelators that we are using for our technology. And it's a pleasure for me also to mention that we have a great collaboration with the clinical experts from Fortress Hospital in India, where a lot of patients have been already treated with Trivehexin. From the industrial point of view, again, just to name a few, we have different collaborations on the let's say chemistry and synthetic part, on the clinical as well as the commercial area. So in principle, once you have the compound and want to bring it to the clinic, there are a couple of aspects that you might not forget. At first is the quality of the compound, so you need to synthesize the precursors in a reliable way. And in this case, on the diagnostic side, we work with Pichem in Austria, and on the therapeutic molecule, we work together with ABX, again our neighbor in Radeberg, which is really a great collaboration, and again with Chematec from France. Once you have the compound, you need to get reliable labeling. You need to have compliant formulation at the end of the day, and here we work together with Eckert & Ziegler on development of the cassettes for rapid and high-yielding production of gallium-68 trivehexin. So this is an ongoing project. Then we work with our distributor BJ Madan in India to supply the hospitals there and have the collaborations also with the academia there. And very important is our partner Radiopharm Theranostics that already has been mentioned by Johannes, because together with Radiopharm Theranostics we are working on the clinical development in the United States, where in very short time the phase 1 with gallium-68 trivehexin will be starting, and we are really happy to be part of this really impressive pipeline of Radiopharm Theranostics. So how to prepare the compound? There are many options. So on one hand, it could be the labeling on the cassette. On the other side, it could be the labeling in a kit, as you see here schematically. The kit labeling is a very interesting approach. It's expected by many centers. Of course, there are different ways. Therefore, we took a look at this opportunity as well, and Trivehexin can be labeled in a single vial approach with different generators that are available on the market at very short time and yielding in a very clean product. There is one topic that is also discussed quite often, and that's the labeling with the gallium-68 produced by the cyclotrons. So as there is the number of the centers that can work also with the liquid targets on the cyclotrons arising, so we took a look at the option to label with gallium-68 from the cyclotron, and there is a good news. It's possible, so you can prepare the compound also in this strategic way, but it's also necessary to point out that even there is a kind of hype about the gallium-68 originating from liquid targets. there is still a lot of development needed, and I believe that the currently available commercially available systems need to be optimized. So there's always some homemade solution in between to make it working also, which we have seen at the site where we were working on this project. And there is a lot of details that have to be considered. So this is something interesting, but we at the moment rely on the production with the gallium eluate from the generators. And before we jump to the clinical part, to the overview, so at the moment we are at the stage where all the parameters have been fulfilled that we need to fulfill till we can move to the clinics. So we know how to prepare labeled trivehexin in a reliable and fast way with high radiochemical purity, high radiochemical yields. We tested, of course, also the toxicity of the compound, which is absolutely no problem in the diagnostic as well as therapeutic setting. And of course, as mentioned, there will be clinical trials now run starting in US. There is a couple of special treatment programs running. This is, for example, this 13-2B paragraph that is used in Germany, and similar programs are possible in different countries. And of course, what we are also supporting are different investigator-initiated trials. So this is also the way how you can approach us and work with us together on this interesting topic. And with this, I'm handing back to Johannes for the clinical update.
So thank you, Jakub, for telling us where to get this stuff. And now we dive deeper into what to do with this stuff. You can see here, I mentioned one of the important indications is head and neck cancer. And here I can show you some data which has been already published. You can access it by this code here. You can see this is an interesting comparison between FDG and Trivehexin in this case. And although I'm not an expert, and you know I'm a chemist and not an MD, there could be a higher selectivity for the Trivehexin here, which of course needs to be demonstrated in clinical studies further. Same situation here. You can see you have many more hotspots here in the head and neck area, and there seems to be an easier diagnosis on this side of the Trivehexin scan. And having that said, there is a study now completed by our very highly esteemed partners of Fortis Medical, and they also focused on head and neck and pancreatic cancer. You can see it here. It's a bit small on this slide, maybe on the screen. Actually, I like this big screen. You can see it really well. You can see that there is a huge difference between the images. Speaking of pancreatic cancer, I would like to point out that this was the first patient we have ever scanned. So this was really impressive to see for us, and really a surprise, I must admit. Here's again some data from India. You can see it clearly here. There's a primary pancreatic ductal adenocarcinoma, which doesn't show up on the FDG scan, but does on the Trivehexin scan. And there's also evidence for the metastatic disease, where there's also some difference in the images. And not only in India, such studies have been conducted. I would like to point out that even at this meeting, a huge cohort of patients was reported. And if you check out which session this was, actually this morning, so if you book it into your calendar, you might be seeing that you're coming late. But I can give you the most important results here in this talk already. You see, here's a box plots of the SUV max in different PDAC lesions, and I think particularly in the primaries, we are in a region which is really giving us lots of hope that there could be ultimately a therapeutic application for this target. I'd like to show you some really impressive cases. This is a pancreatic primary, although it's not a ductal adenocarcinoma, which has a really high uptake. And again, here's a liver metastasis, where you can see this is also an uptake which definitely calls for therapy. And of course, you've seen that there is a large spread of the uptakes which means that we have to consider this, of course, in our concept, how to advance this asset towards the benefit of the patients. So imagine you have a cohort of patients with unknown alpha beta 6 status, and what we can do already now is to use our established diagnostic Trivehexin to filter out the cohort which expresses alpha beta 6 integrin. And then it's obvious, this is what most part of this meeting are all about. We go for therapy, and we are about to develop this therapeutic ligands. And this is, of course, not the end of the story. The same target can, of course, be used for radio-guided alpha beta 6 guided fluorescent surgery. So there is, particularly in terms of head and neck cancer, it could be really interesting to have a clear intraoperative image of the dissection margins, which could help the surgeon greatly in assessing where to cut and where to leave the tissue in the patient. And there's another aspect, which is targeted drug delivery with drug peptide conjugates. Of course, if you have a target which is highly expressed and internal on tumor cells and internalized by the tumor cells, you have the potential to greatly advance the toxicity and effectivity profile of a lot of cytotoxic drugs, which without using such constructs are not amenable for therapy in humans. And then we have the general benefits of doing PET imaging for preventive screening or simply wait and see in some stable disease, and of course also for classic surgery approaches just in one sentence to question whether or not to perform surgery and decision on the image. And with this image, I would leave you, and I'm happy to answer questions, and of course, Jakub also.
Thank you very much. I would like to ask all the speakers of the second part on the podium, and if there are questions from the audience, please don't hesitate. So if there are no questions, let me please ask you one question, Johannes, because I stumbled over the babies in your last slide. Do you intend to image babies, toddlers?
Well, this could be taken as a not serious approach, but actually I'm totally aware that pediatric PET imaging is probably not what you would do every day, but consider that there are total bodies and whole body scans around. And the normal imaging dose we use for Trivehexin is 150 megabecs, and we end up with 4 millisieverts. And with those highly advanced, highly sensitive scanners, you can go to 5 to 10% of this dose, and then you end up with something like 0.2 millisieverts, which is slightly more than a breast CT. So I think, if we as a company, we have to think not about today's situation, but about the situation in 5 or 10 years notice. And in 5 or 10 years, we can do all the imaging with much lower doses, and therefore address a wider patient collective and also minors, of course.
Thank you. Andreas?
I have a question to all of you. So you all have the approach to treat pancreatic cancer, which is really very difficult. And when you use radiation, there is some fear from internal medicine physicians that we cause pancreatitis when irradiation is going on in the pancreas. Did you consider that, or do you think we can just use it similar to PSMA or CXCR4?
I would like to clarify with a question from my side. Do you mean that the pancreatitis is treated?
No. It's a side effect of your treatment, because the pancreas is also sensitive to radiation. We have a lot of enzymes around, and when you have beta decay in there, you could make it happen that pancreatitis or very severe pancreatitis is going on.
Since I have the microphone, I will simply tell you my answer and then hand over.
Sure.
I think since we have a very selective target, and particularly in the pancreas, we have an extremely low background. And we have more background in other organs than in the pancreas. So one could, if the beta radiation is too, the range of the beta radiation is too long to cause severe pancreatitis, one could probably circumvent this with doing alpha radiation, which really kills only the tumor cells and leaves the rest of the pancreas in peace.
Yeah, I mean, not a lot to add to that. And to be fair for us, I mean, FAP as a target, I mean, we are in the, I mean, pancreatic cancer, of course, is very relevant, but we go beyond that. And so today we are still in the phase of selecting indications for a first clinical trial. I think only time will tell what will happen, what is the risk of treating or at least targeting pancreatic cancer. Yeah.
Thank you.
Thank you for the talk. I have one quick question for Precyrex. I'm interested in the background to you, why you were choosing iodine-131 as an isotope.
Yeah, that's a very good question. Because we all understand that the choice for iodine-131 is not the most logic one. I mean, I think also here the majority of the talks would be lutetium, would be actinium-225. And so for us, it all started with a particular observation, back in the academic days that combination with halogens has a major beneficial effect on the biodistribution, on the in vivo pharmacokinetics of the compound. And then really focusing on what happens upon renal filtration. And so we see that when combining with iodine-131 and combining with particular ways how to conjugate your isotope to your single domain antibody, that we have, as shown here, both preclinically but as well as with clinical evidence, that we have a very beneficial profile and that we do not see any relevant retention in kidneys upon filtration. And of course that opens up the possibilities. You know, you have a therapeutic index that all of a sudden ranges between 5 and 10, whereas potential combinations with other radioisotopes and means to conjugate isotopes to the single domain antibody, that therapeutic index is less optimal. So that was really the driver for iodine-131. Yeah.
Can I ask a quick question as a follow-up? What's the labeling strategy you use? Do you use prosthetic groups?
Yes, that's correct. Yeah, yeah. And in fact, it's all published work. So that originates, in fact, from an back in the days academic collaboration with the lab of Michael Zalutsky, Duke University. And so we together identified one of his very particular linkers. Yeah, absolutely. And so it gives rise to fast clearing catabolites upon filtration. And so that was really the key thing. Yeah.
Okay. Thank you very much. I think for the sake of time and also because we have to clear the room, we're going to have to come to an end of this symposium.